Hello, everyone. I'm the Chief of Pediatric Neurology here at UT Southwestern, where we're building a big gene therapy program. Steve Gray was recruited here, and along with him came Rachel Bailey, who's just been fantastic. And the work she's doing in SLC13A5 is, just very, very gratifying. I am in the middle of multiple emergencies, so I will be brief and I will not use slides. I'll just give a general sense of things, and honestly, slides are not needed. I'll stay as long as I can, but I apologize. We have 30 faculty, and although we haven't been hit yet, we are in the midst of lots of work to try to prevent children from having this problem. Okay, so the question is to do a gene therapy clinical trial. What we're talking about is basically replacing the missing gene in these kids. And this project, SLC13A5, is one of many that we've picked up. And Rachel has really taken it to heart. I feel you have a contagious problem yourself, Kim, and Rachel, Brenda Porter, and others I've met who have jumped into this disease are just so wonderful and just so engaged. I must say that although we're very engaged, this is one of the more difficult projects that we've taken on. Firstly, there's no appropriate mouse model, and there's a number of other issues. But we will keep pushing, all of us, until we get there. For a gene therapy clinical trial, you can do it in two ways. The ideal is to do it in such a wonderful and perfect way that the FDA would see results showing efficacy and obviously lack of toxicity, and it would be so clean and neat and well done that they would allow registration of the quote-unquote drug, meaning the viral gene therapy, so that then it could be prescribed by doctors for future patients. And obviously that's by far the best goal. But the standards that one has to meet to have a registration enabling study are very high, and I'll go a little bit into that. The other approach is to simply do a safety study, which is kind of a typical approach for any drug development. And then there you're not looking to prove efficacy, but simply to show safety. And there the standards are lower. But the problem there is that you cannot register the drug. So the few patients who participate in that safety study would get the gene replaced and may benefit, but there would not be enough rigorous data to convince the FDA or anyone that this can be marketed to the population at large. Why did I make this distinction? Because it's very important on how we design the study. Natural history has been discussed yesterday, and I'm not sure to what degree today. What is that? Well, any disease has a clinical course. It starts in a certain way and then progresses in a certain way. And when you have enough patients, especially if the patients more or less have a similar course, then you can say this is the course of the disease. So to prove that your treatment, whatever it is beneficial, you need to show and document and have actual quantitative data, quantitate the differences that happen to the patient who is treated versus a patient who is not treated. Normally, for a normal drug study, the approach that is needed for registration enabling is a double-blind, placebo-controlled study. So some patients get the drug, and the other patients get something that looks like the drug, feels like the drug, but is sham, basically, does not contain the active ingredient. This cannot be done in our gene therapy trials in rare diseases for two obvious reasons. Firstly, the patients are very rare. Secondly, it's really hard to see how one would justify injecting something or whatever into patients, you know, with such terrible diseases, and it would be a sham injection. So therefore, since a placebo-controlled double-blind study is very difficult, if not impossible, to do in these ultra-rare diseases, the FDA has permitted an alternative. They say, if you can quantify and properly delineate the natural history of the disease over a number of years, and then you can take that curve I'm showing with my hands, the curve of how the disease progresses in all the elements that you quantified, whether it's motor or neuropsychological or the number of seizures or all the different things that you can quantify. And then when you actually do your study with the drug, at least you have a comparison arm to which you compare the results of your study. And then you can quantify the effects on your study population and compare the quantities between the natural history curve and the treatment curve. And then you would have statistical power to show that the drug really made a difference. This is why it is essential to have this natural history study and to have it done well. The FDA considers the natural history study as the control arm of the eventually potentially registration enabling clinical trial. Therefore, the data collected in the natural history study should be done with extreme rigor. And I'm not just saying these words like extreme just to say a lot or something. We've come to learn how extremely rigorous these data must be collected for them to be accepted by the FDA as meaningful. So it's very, very important to do the natural history as well as humanly possible, actually within certain guidelines established by the FDA. So when we get to be doing this, our team knows very well what these types of criteria are. Dr. Porter is extremely experienced and the others involved. So we will all make sure to put our heads together and not do a study that may end up not being as useful as it could be. So now, SLC13A5. This is a special disease. I don't know the disease well enough. I'm continuing to try to learn. I tried my best yesterday, but I had to be pulled out. But I'll keep on trying to learn. I'm very happy that Dr. Porter knows the disease well, Dr. Goodspeed is knowing the disease well, and that there are such excellent families and parents who can teach us. Now, if you think of a natural history in the traditional sense, you take a neurological disease in an adult, for example, Alzheimer's disease or Parkinson's disease. Those are people who have been healthy, and then they have neurodegeneration of the brain. So there's a very typical course of events that happen over time in the Alzheimer patient or the Parkinsonian patient. So it's very easy to delineate the natural course of those diseases and have a curve against which to compare any drug treatment. It is not the same in pediatrics. Indeed, in pediatrics, we do have diseases where the kid is normal and then neurodegeneration happens. But pediatrics, by definition, is the period of development. And the human brain during development, during childhood, is very different day after day after day. We're not dealing with the same kind of constant brain that might be something going wrong in it. We're dealing with a moving target. The brain is trying to develop and is developing. Meanwhile, there's a disease happening to it, which is leading to problems. So we have a particularly difficult situation in a neurodevelopmental disease because there are two moving parts. There's the natural development of the brain, and then there's the movement of whatever the disease is causing. In SLC13A5, from everything that I've gathered, it's particularly complicated in that we don't have a period of total normalcy and then degeneration. We have this kind of ongoing mix of the effects of the disease with the effects of development, making doing a proper natural history study that much more challenging, which is why it's been really complicated to figure out precisely how to do this. But I do believe the best minds, you know, in the field have come together, and we will definitely do it as best as humanly possible with the leadership of Dr. Porter and the contribution of all the other neurologists involved. I have one last thing to say, and then I'll have to go. I'll see if I can take a burning question. I don't know if you've been doing questions. The one other thing to say is that the FDA has also weighed in and said, if there is a biomarker that you can find which correlates tightly with the clinical condition. So for example, let's just say, take any disease X, and then there's a blood test or a lumbar puncture test that you can do and measure something. And if whatever happens to the patient always correlates to a change in the number of that test, the blood test or the CSF test, then the FDA has said, if those two things are totally linked with each other, you can use the biomarker in lieu of or in addition to your natural history study to track. What that means is, I'm just going to take the word citrate here, but you will forgive me, I couldn't participate in the whole meeting and a lot of it escapes me. But let's just assume, for just discussion's sake, that citrate level correlates very precisely with the neurological phenotype of SLC13A5. And if the citrate is corrected, the neurology is better. And if the neurology is worse, the citrate is worse. If there is a connection like that, then you can use the biomarker instead of or in addition to your natural history, because they go hand in hand, which is the reason why this citrate aspect needs to be explored maximally, and these connections made as much as possible to help have the tools we need to prove efficacy. If we cannot prove efficacy, then we cannot have a drug that goes beyond the clinical trial, meaning beyond the few patients who have received the treatment during the clinical trial. I will stop here, and if you wish to take questions, I could take a couple of burning ones, but then I'd have to go to my other meeting, and I apologize for the shortness of the time.
That was great. Okay, we'll break the rules here. If people have a question, they can either quickly type it in the chat box, or you can unmute yourself and ask Dr. Minassian your question.
This is Brenda. So I totally see what you're saying, but is it appropriate to go to the FDA and say to them, you know, we have this sort of disorder that doesn't have well-characterized biomarkers? Would you give us guidance at this point on making sure we're collecting it in a way that is appropriate and, you know, enough data? Like, how do you get them to weigh in at this point? Is that even possible?
Yes, so we've had, given the number of diseases that we're dealing with, we've dealt with the FDA a lot, and it varies a little bit how they respond. It's almost a case-by-case basis. Sometimes they're very flexible. In a recent similar situation, they were rather rigid, and we had to do all sorts of other experiments. It was another situation where there wasn't, where there isn't a mouse model, and they simply, you know, would not support moving into a clinical trial based on mouse results because the mouse did not have a phenotype. So we had corrected something in the mouse that we could test neuropathologically, but they said that that's not appropriate to give us permission to go to clinical trial because there's no connection between that neuropathological change and the phenotype because the mouse completely has no phenotype. So in this case, you know, we're trying to generate a pig model and find other approaches. Rachel knows a lot about this and can answer more questions when I step out. So definitely we can interact with the FDA, and we will. They're certainly kind and helpful, but more recently in this particular case, they kind of insisted that we had to do something else and more.
Any other questions?
Thanks.
Otherwise, I want to make sure we get to Rachel before it gets much later. Okay, thank you so much, Dr. Minassian. I really appreciate you joining us.
Thank you, everyone. Stay healthy and
Bye.
talk soon. Bye-bye.
Bye.